Our next speaker is Javier Sancho. He is full professor in biochemistry and molecular biology, and he is the director of the BFI Institute at the University of Zaragoza. He has promoted within BFI the creation of LACRIMA, that is the Aragon Drug Discovery Center, and he is the leader of the Protein Targets and Bioactive Compounds Research Group. And this group brings together expertise in drug discovery, protein engineering, and biocomputation. And as well as all this, he is also interested in technology exchange and in advanced teaching. And today he's going to talk about precision compounds to fight infectious and molecular diseases.
Thank you very much. I think I can skip the first slide because you have perfectly introduced what I wanted to say. So I will begin by introducing the center where we are working, that is the BFI Institute, which is 1,500 square meters devoted to biocomputation and drug discovery. Around 100 people work there, 60 of them are PhD, and we have the fortune to enjoy of the largest computing center in Aragon, the Center for Supercomputation in Aragon, and also the LACRIMA Drug Discovery Center that was already mentioned. In this center, we try to obtain from scratch, focusing on a specific target, novel compounds, novel families of compounds with bioactive properties, and we are using this capability to try to develop drugs. We are also interested in drug repositioning. Our approach is to deal with the problem and the opportunity of precision medicine. All of us are different from the others because we carry from 4 to 5 million genetic variation from the standard sequence of the human kind. And in addition, the bacteria that infects us along our lives introduce in our bodies an extra variety because some of them may be resistant to particular antimicrobials, etc., etc. So in some cases, we have the need to discover specific drugs, even for diseases for which there are some existing drugs that are not good enough for the whole population. Our approach is to begin with a chemical library. We have chemical libraries. Of course, we can work with chemical libraries of other people. And then we set a specific screening test devoted to find the compound that targets our target, that is either protein or a bacteria that we have to kill. So we work very much in devising the best test to perform target to hit or phenotype to hit screening. And then we use our own analysis methodology, our own scripts and programs to rapidly interpret the data. And after doing this screening against a particular target, we get a hit, and then we embark upon medicinal chemistry. When we have the structure of the complex between the molecule, the small molecule, and the protein, we try to do rational biocomputational methods to improve the compound. And when we haven't that structural information, we hope that our serendipity will help us design new changes in the molecule that we test, and in that way we improve the properties of the initial hit and we make a lead. And in that moment we test the compounds in animal models, in many cases collaborating with specific groups that have the specific animal models that we need. I will refer now, since there are several speakers in this audience that are working in LACRIMA and are developing specific projects, to 3 projects of my group, the Protein Targets and Bioactive Compounds group. The first one refers to developing of novel antimicrobials to fight infectious diseases, and I will refer to the infection by Helicobacter pylori, which is a large pandemia. Actually, in China, there are around close to 800 million people that have this bacteria inside their body because half of the population in China, 56%, is infected. To make the story short, at this moment we have a family of compounds. That can eradicate the bacteria in around 20–30% of the mice we treat. This infection is treated by a combination of 3 or 4 drugs. So we are trying to develop drugs that on their own individually can target this disease, and we are halfway of having something already good enough, but perhaps it can also be considered to be used in combination. The second project deals with pharmacological chaperones, that are small molecules that bind to proteins that carry mutations that impair their function. In this project, we are dealing with Phenylketonuria Pku, which is a rare disease, but in China it affects to over 100,000 people. This disease is caused by many different mutations, more than 1,000 mutations in this enzyme that transforms phenylalanine into tyrosine. And this disease, if untreated, it is treated with a very strict diet, cause severe neurological disorders. And there is a drug in the market that ameliorates the conditions of around 1/3 of the individuals that suffer the disease. And what we have done is to develop several families, several different chemical families of compounds that we are testing on the mutations that are not properly treated by the existing drugs. So we have a family of cells lines, and we are testing our compounds on the lines that do not respond to the existing drug. And the third project I would like to talk about refers to inhibitors of amyloid aggregation. We have worked in several diseases, in Alzheimer's and type 2 diabetes, but the example that I bring to your attention today refers to Parkinson. This is still unpublished. And Parkinson is a disease that affects around 2% individuals in the world, over 27 million in China. There should be the Parkinson here rather than PKU. And what we have done in Lacrima is to discover one compound that has been tested by our collaborators in the Autonomous University of Barcelona using, hopefully I can, yes. Oh, they won't move. I will explain. This is a movie. This works. Oops. Can you click on the 2 panels, perhaps? Click on them. Can you? No? Okay, never mind. The worms on the left-hand side are not moving. They are a Parkinson model, and the worms in the right-hand side are very happy moving around because they have been treated with our compound. So we can cure Parkinson in worms so far. It's okay. Try? No? Okay, leave it like here. So the summary of this talk would be that if you have a target that you for which you want to find out a novel drug, we have set up all the labs and all the equipment and all the methods that allow you to go from the target to compounds that are approaching the end of the preclinical development phases. So you just need to know what is the protein target or what is the bacterial target that you want to target, and we can identify promising compounds that will be close to entering into clinical trials. We are eager to collaborate. We are very happy with new proposals and with discussing how the technology can be applied in other places. And I have reserved a final slide to mention very briefly 2 things. One is that in many cases, novel drugs are not small molecules, small organic molecules, but proteins. This is the case of biologics, enzymes, monoclonal antibodies, etc. Proteins are much more fragile, much less stable than organic compounds, and quite often, they pose a problem of stabilization. This morning, in several talks, I've heard that in some cases, the companies were happy that they had stabilized their proteins. We would like to mention that in addition to using formulation to stabilize proteins. Proteins can be thermostabilized through protein engineering by making point mutations, and that we are experts in this technique, that we believe is very valuable for those that are interested in developing proteins as drugs. If your proteins are not stable enough, we would be pleased to help and stabilize your proteins. And finally, we also believe that there is a training aspect to the drug discovery process, and last year we set up a new master study in quantitative biotechnology, which is very much devoted to teaching students how to develop drugs from scratch, how to stabilize proteins to make them into drugs or into valuable analytical tools. And this is all. Thank you very much.